Welcome to Unit 4. Unit 4 is all about ecology. So we're going to cover a lot of the basics that you've probably touched on in previous science classes, especially, I know for a fact, you covered this in your freshman bio class. We're also going to talk about energy flow through systems. We're going to talk about cycles. We're going to touch on a number of cycles, but focus most of our energy on the carbon cycle, and we will end this unit discussing climate change. So, like I said, this unit is probably stuff that you've already talked about at some point in your science career. A lot of this entire presentation is going to be vocab and kind of reminding you what these terms mean. So I'm not going to go through everything individually, but I would suggest maybe pausing and writing this down, or if you feel super confident and like you don't need to write it down, that's fine, but just know that these terms will absolutely show up again and will absolutely show up on tests and quizzes. So, brief overview, talking about ecology. In this sense, we're talking about groups of organisms or singular organisms going from small to big, right? A species is a singular group of organisms like just the crab, whereas if you go all the way down to ecosystem, that is everything that you see in the picture, right? All living and non-living things. Ecology is kind of the oddball on this slide. It's not a type of group of organisms, it's just the study of how organisms interact with each other and with their environment. So that is the definition that we're going to use moving forward of ecology. So talking about types of relationships, we've got some positive relationships, which I know you don't think predator-prey is positive, but they are intertwined with each other, so it is actually a positive relationship. I'm sure the bunny in this graph does not feel like it's a positive relationship, but one organism hunts and feeds on another, but if the rabbit population were to decline, then the fox population would also decline because it cannot survive without the other. Symbiotic relationships, I know you've seen this, so I'm not going to go through this, but be aware that these are coming up again. Mutualism, commensalism, and parasitism. Mutualism is two benefits. Commensalism is one benefits and one's like, meh, I don't care, cool, I'm along for the ride. And then parasitism is one benefits and one is harmed. For niches, we're going to get pretty deep into niches, so there's a lot of pieces that make up a niche. So one is just the habitat where it lives, whatever organism we're talking about. So if you're talking about like deer, you would say that they live in the woods or the forest. The activity patterns of the organism, this doesn't mean that like it likes to cross the road at 1 p.m. Like, what does it typically do in general? So it's always going to feed in this certain area, or it's always going to sleep with X amount of other deer for protection, right? Number 3, resources it obtains from the environment. So what it's eating, where it goes. If it doesn't go beyond, a certain river, that's part of its niche. And then interactions with other species. So whether it's competing for food, whether it's being hunted by other organisms, just things to keep in mind. So there are types of niches. Again, like I said, we're going to get pretty deep into this. So there are 2 types of niches, and then there's differentiation among that. So the 2 types of niches are fundamental or realized. A fundamental niche is where it can live. So any area in which, again, I'm going to use deer as my example. So any area where a deer could potentially survive. So this is probably much larger and much different than the realized niche, which is where it actually does live. So fundamental is kind of the broad, vague, yes, I could live here. Yes, I could survive here. But realized is what's actually happening. So realized is what you would study when you are studying a certain organism. And then niche differentiation is talking about how organisms of different species interact with each other. So there's competitive, and then there's resource partitioning. So competitive is exactly what it sounds like. One species is going to compete with another species to use the same resources. Typically, this involves one species, quote unquote, winning and driving the other to extinction. Not full extinction, local extinction, where it cannot survive in that niche. And then resource partitioning is that the two species will change what they typically use in their own niche to avoid direct competition, which allows them both to survive in whatever realized niche they're in. So to show that in another way, again, I just said this verbally, but I personally like pictures, so you'll see that I use pictures to kind of really emphasize whatever point I'm making. So on the left, where you see grown separately, you see that there's no overlap because they're not even remotely involved with each other, right? They're in two separate test tubes. But then when they're grown together, you have the two versions of the niche differentiation. So competitive, you'll notice that the blue one the blue paramecium took over and drove the pink paramecium to local extinction, right? It does not exist anymore. It's only the blue one in that test tube. Whereas resource partitioning, they've split it so that the blue is on the bottom and the pink is on the top, and they do not interact with each other because they're using different resources. These are, again, probably pretty straightforward. You've heard these terms before, modes of nutrition. Autotrophs feed themselves, right? That they go through photosynthesis. Some are also chemoautotrophs, meaning that they make organic compounds using energy from chemicals. Some ocean-dwelling organisms do this. And then there's heterotrophs, which is like you and me. We have to eat other things to get energy. And then heterotrophs can be broken down even further. There's consumers, detritivores, and saprotrophs. And we'll get more into those three in the next couple of slides. So metabolism is classified in three kind of key ways. I'm pretty sure you covered metabolism last year, but just to be super, super clear, metabolism is the idea of your body converting whatever it is that you're eating or absorbing into energy. So there are some carbon sources, right? So autotrophs get their carbon from inorganic compounds, heterotrophs get it from organic compounds, and then mixotrophs get it from either source. And then there's energy sources, so photo or chemo, where this is kind of going back to the two different types of autotrophs. So you're either getting it from sunlight or externally derived chemical compounds. And then reducing equivalents. So this is how you're getting your electrons. So we are getting our electrons from organic compounds, and then there are other organisms that are getting them from inorganic compounds like bacteria. So they're getting it from like ammonia or sulfide or iron, different ways, but they could get it from like the soil or from the ocean. So moving even more in depth with autotrophs, you've seen this, you know this, so I'm not spending a lot of time on these things. So autotrophs gain their nutrients from abiotic environments, typically the sun, from using photosynthesis, and then heterotrophs obtain these substances from eating other things. So either eating autotrophs or eating other heterotrophs. And then heterotrophs, this is kind of what I was talking about, that they get divvied up even further. So there's consumers, herbivores, carnivores, omnivores, depending on if you're eating only plant matter, only animal matter, or either plant or animal matter. There's scavengers who eat dead and decaying carcasses, right? Like a vulture or a hyena. The detritivores, which obtain nutrients from fecal matter, like earthworms and dung beetles. And then saprotrophs, which secrete digestive enzymes into non-living matter and then break down those non-living items. So it could be like dead trees. They excrete their enzymes, it breaks it down, and then they absorb the products of that digestion. So that is like bacteria and fungi. That's why we call them decomposers. And then this is like a super brief overview. We're going to go way, 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 way more in depth with nutrient cycling, but this is the idea that nutrients are limited. And so constantly they are being recycled so that they can be recreated and reused, because you can't simply just poof more carbon dioxide or like poof more phosphorus. It doesn't work like that. They are a limited substance. Okay, talking about sustainability, typically ecosystems are self-sustaining. A lot of the ecosystems, if man did not interfere with them, would be fine and they would self-sustain. The three main components required is how much energy is available, how many nutrients are available, and how the recycling of waste happens. So if there's enough light from the sun, then the energy availability is going to be fine. If the nutrient availability, if saprotrophs can decompose to get that recycling of these nutrients back into whatever ecosystem they're in, that's going to be okay too. And then getting rid of harmful waste byproducts, you need to be able to maintain that so that there's not a buildup of these toxic waste, so that you're not losing area or food or shelters that you might need. Okay, this is the last bit really for this presentation. We're talking about quadrat sampling. So this is when you're kind of determining the presence of species in a given area. So you specifically make a quadrat, which is like a big square, and you measure what would be found within that quadrat. So a positive association is if you find two species in the same habitat, and negative is if they don't tend to occur. You usually use this for things that don't move, right? It's pretty hard to be able to measure how many deer are in a quadrat when deer just constantly move. So this is typically for things that like are plants, or even things that rarely move like fungi or other more immobile animals. So you can see the different types of sampling distributions, whether it's even, random, or zoned, meaning that you have small clusters within a certain quadrat. And then that was on the right picture. And then on the left, you can see how you would count. If something is mostly inside the quadrat, you do actually count it. It doesn't need to be fully in, it just needs to be mostly in. And then the very last thing, still in relation to quadrat sampling, is chi-squared tests. This is a statistical analysis that I think works really well for a lot of IAs. So we'll get a lot more into this when we cover our statistics unit, so that you know what you should be doing and what you should be using for your IA. But I'm just going to briefly go over this, and we're going to do a few practice problems, but we're going to do a lot more practice problems next unit when we get into genetics. So the main thing here is you're trying to determine if there's a statistically significant association between two species. So this is not always used. It's not always the best statistical analysis to use on an IA, but if you're comparing two different samplings of something, it can be really beneficial. So an example of this could be determining if you bought a bag of Skittles, if you have an equal chance to get a red Skittle as you do to get an orange Skittle. So you're kind of seeing if there's a significant association between the types of Skittles. So you have to identify hypotheses, a null versus alternative. A null is always going to basically be saying that the variables don't interact with each other. So again, in this case, you could say that if we're still talking about the Skittles example, that all of the candy is evenly distributed and there's no significant difference between the colors and flavors of Skittles that you get in a bag. Whereas the alternative would then be the opposite of that, saying that there is a difference and no matter what Skittles bag I get, I somehow get way more orange than red. And then you construct a table of frequencies. So in this case, if you had the 5 colors of Skittles, you would assume that the expected would be equal amounts of each of the Skittles. The observed would be literally what you count. So it would not be exactly what the expected is. If it is, then that's crazy, but it's not typically. Then you use a formula, and we'll get more into this when we start practicing with it. And then determining the degree of freedom is based on how many different options you have. So how many types of colors you have, minus 1. And then the p-value. This is given to you. You'll never have to memorize what p-values are, but you should know that it should always be less than 0.05. That if it's less than that, then you can say that you accept the alternative, right? And that there is a significant association. And if it's more than that, then you have to accept the null hypothesis and that there is no statistically significant association. Again, we're going to get way more into this, and math is hard to explain via video, but when we start doing actual practice problems, it's going to make more sense. So again, this is just kind of an overview of what chi-square tests are, and we'll cover it in more depth in class. All right, thanks for tuning in.